Hey everybody, this is Miss Segovia and I'm going to walk you through the dihybrid cross. So sometimes in genetics we won't look at just one trait, but we'll look at more than one. And so in a dihybrid, since it's the word di, we're actually looking at two. So in our sample problem we have here, in pea plants, the green color allele G is dominant over the yellow color allele lowercase g for seed color, and tall, uppercase T, is the dominant allele in a plant height. Parents heterozygous for both traits are cross-pollinated. Determine the frequency for the four different phenotypes of the offspring. And so what they mean to say here, since they said parents are heterozygous for both traits, that means that they have both the uppercase and lowercase letters, both the dominant and recessive. So that means one parent is uppercase, lowercase tall, and uppercase, lowercase green, so they'll show as tall and green, and both parents are that way, so we would have them crossing like this. Now there's two different ways to do this. One is the 16 square punnet. I'm going to walk you through that one really quickly, but then I'm going to show you a much easier way that I think you'll definitely enjoy. So to set this one up, we have to look at what are the possible gametes that the offspring could receive. And so it is possible that they could receive the uppercase T and the uppercase G, the uppercase T and the lowercase g, the lowercase T uppercase G, and the lowercase T and lowercase G. Sorry, it's a mouthful. Since the parents are both heterozygous, it could be the same from the other parent as well. Now I could tell you from years of experience that when kids do a cross using this type of table, they tend to screw it up. Sometimes they'll put like just the T's and then just the G's, or some weird mixing of them, or 4 and the end of the 8, and it's just a mess. But you work this like a regular punnet, except that each offspring would have 4 genes instead. So you would have uppercase, 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 uppercase. So that would be a tall and green offspring. Here we'd have upper, 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 lower, still tall and green. And you just kind of keep going. So I'm going to fill this out real quickly, and you can go ahead and continue doing that as well. And I think it's better for us both if I just stop talking, because it's hard to talk and write at the same time. Oh, here we have a tall yellow. And notice I'm putting both the genotype and the phenotype in here. That's going to help you later on. And this one, we ended up with 2 lowercase t's, so this is our first short one. That's a short green. Here's another short green. Oops, goof that up, it should be lowercase. Yeah, so that is a tall yellow. Now keep in mind in Punnett squares, this doesn't mean that they're going to be having 16 offspring every time they mate. It's just giving us the odds of what the probability of an offspring would be from this mating. So now we got to count up what we have. So the number of plants that are tall and green. And so 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah. So we have 9 out of 16. Tall yellow, we should have 1, 2, 3. So that's 3/16. Short green, we have 1, 2, and 3. So for 3/16. And then just 1 short and yellow. And see, this process takes a lot of time and it's easy to goof up, and it's just a pain. So there's a much easier way to do that, which is doing a little bit of math. So let's take a shortcut way of solving the same dihybrid cross. Because of Mendel's second law of independent assortment, you can work with the color gene and the height gene separately. So set up two separate monohybrid crosses from these same parents. Now, for those in advanced genetics, this only works if the genes are not linked, if they are not on the same chromosome. If they are on the same chromosome, then it's a whole nother set of rules. So basically what we do though on this one is we just do mini punnets. And so we look at just the height gene. So we know that we had heterozygous parents, and so we would draw out the punnet for that. So here we've got, this is tall, our phenotype, tall, 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 and this guy is short. And then we also work out our color gene. So big G, little g's, that's green. Big G, little g, that's also going to be green. Big G, little g is green. And little g is yellow. So now that you have this and you have the probabilities of each one, we go down, we do just a little bit of math. And it's easy math because it's multiplying of fractions. So our tall plants, what number in fraction do we have in tall? And so 1, 2, 3 out of the 4. So we put 3/4 here for height. Our color is green, and so 1, 2, 3 out of 4. So that is 3/4. And then we multiply. 3/4 times 3/4. Well, 3 times 3 is 9. 4 times 4 is 16. Oh, didn't we see that before? Tall green, 9/16. Why, yes, look at that, 9/16. We get the same answer. Let's try it again for tall and yellow. We still have 3/4 of our plants being tall, and in yellow, only 1/4, 25% chance. 3 times 1 is 3, 4 times 4 is 16. So if we look back up here, yes, it still matches 3/16. Do the last couple ones here. Short plants, that's 1/4. Green is 3/4, so that is 3/16. And then short and yellow, 1/4, 1/4, 1/16. And so you can see the numbers here match the numbers we got back in the big table. But as far as level of difficulty, I personally find this method much easier. And you can also use it if you have more than just 2 genes that you're looking at. You know, the number's really infinite using this method versus having a table that would just get exponentially larger and larger and larger. Not any fun. By the way, a typical dihybrid cross where you have heterozygous parents, the ratio that we see in this is 9:3 to 3:1. So in advanced genetics, that's a very key indicator when we're looking at our numbers of offspring. So there's a foldable that goes with this, and I'll shoot another video to help you answer those questions. Thanks.